ridge structure to it. When it's fully effaced, the next thing that's going to happen is with the pressure of the head, the cervix is going to begin to dilate. Okay? And so when you're kind of getting into active labor, you're going to probably, when you do the palpation, you're going to find that the cervix is fully effaced and maybe 3 or 4 centimeters dilated. At that point, that woman is probably in active labor. Okay? Now, the sequence of events that actually has to occur, it has to happen very quickly here, is that when the baby's head now is pushing against the cervix and it's softening and effacing, you're basically going to get some neural impulses that go up to your, that basically go to your hypothalamus, your posterior lobe of your hypothalamus. And then when the cervix begins to stretch, the posterior hypothalamus is going to release a very potent hormone called oxytocin, which is also known as Pitocin. Okay? And this causes very regular, firm, forceful contractions that occur every 2 to 3 minutes. And this is what propels the baby through the birth canal. So what I'm showing you here is a picture where there are a whole bunch of hormonal events that are timed together that have to happen in the correct sequence in order to end up with a normal labor and delivery. And it's also true that, if a woman has, for example, has ruptured membranes, okay, if the membrane's ruptured, you have 12 hours basically to deliver the baby. You might want to speed things along. And so if you have ruptured membranes, you go over to the hospital, you're going to probably get put on a Pitocin drip, which is going to augment the contraction of the uterus so that you can get really productive labor going quickly. Why is that important? Why does all this have to happen in a rapid sequence? Yeah, that is a fact. If you have a really long, prolonged labor, you have to worry about sepsis. And that's a dirty place now. Okay. And so you can get bad bugs, you can get into your bloodstream and cause sepsis, which is actually the leading cause of postpartum death in women. Sepsis. But why does this have to happen very quickly? Why do all these events have to happen quickly? Yes?
The muscles get fatigued.
No. Think about the poor baby. When the uterus contracts, there's no blood flow to the placenta. Okay? And so the baby is going to, with a prolonged labor, is going to become anoxic. In fact, you can have, the poor baby can have a stroke. And actually, when that happens, we call that cerebral palsy. Have you ever heard that? Cerebral palsy just means bad labor. Baby didn't get out in time. There's a race. Baby's got to get out. Because this is a really, really tough time just in terms of the blood flow. And so one of the things when you're in the labor and delivery suite that us biomedical engineers have really helped with is you're going to get the fetal pulse monitor, usually the ultrasound. And so what happens when the baby becomes anoxic? The fetal heart rate goes way up. I mean, ordinarily the fetal heart rate is 150, 180. Boy, it goes way up when it doesn't have enough oxygen. And that tells you the baby's in distress. Another reason why sometimes you're going to have to speed things up. Okay, any questions? Okay. So, we biomedical engineers have all kinds of ways that we can measure electrical activity of the muscle. So we have little needles If you come over to my lab, I can show you some little needles that we can actually stick in your muscle, and we can measure individual motor units. We have just kind of a hypodermic needle that's insulated, but we can measure just the muscle we stick the hypodermic needle in. We can do a concentric needle EMG, or we can actually do a single fiber EMG, depending upon how we kind of arrange the electrode and how we get the differential amplification of the signal that we want to look at. And so we use this actually to diagnose different kinds of muscle diseases. Probably the most common thing that we do is just a surface EMG. And y'all may have done this in one of your undergraduate labs. Did y'all do an EMG? Or record that in one of your labs. Anyway, you just take silver chloride, 2 silver chloride electrodes with the ground, and you can measure these. You can actually measure these huge, really huge compound action potentials on the surface of your skin that correlate with the strength of the muscle contraction. And so, when you contract, you'll see a very large amount of activity, electrical activity. And I want to point out to you that this scale here is 10 millivolts. We're measuring 10 millivolts on the surface of your skin. It's just measuring how much your muscles are making action potentials. And we use this as a way to kind of judge the amount of force that muscle is creating. It's not really kosher to cut a muscle and put a transducer in series with it. So we actually use this as a proxy for judging muscle contraction force. And so it turns out that if you do the integrate and you do the root mean square, so basically I integrate this over a period, so I do 1/2 integral from 0 to t of v squared dt, and that gives me what's called the root mean square, the RMS value of the power that's in this EMG signal. And this correlates with the log of the force that you're actually contracting. And so there's actually an experiment that shows that you can pretty much predict how much force that you contract just by looking at the energy in the EMG signal. Okay. One of the diseases that has been in the news pretty recently with the ice bucket stuff has been ALS. Okay, y'all follow that? You know what ALS is? Y'all know what amyotrophic lateral sclerosis is? Yeah. Y'all know anybody who has it?
Lou Gehrig.
It's Lou Gehrig's disease. Lou Gehrig passed away a little ways. Yeah. So it usually affects folks when they're in their mid-50s to 60 years old. Once diagnosed, you have about 4 years to live. You basically have perfectly normal cognition. This is a combined upper and lower motor neuron disease. So you get muscle wasting and muscle weakness. You have both upper and lower motor neuron affected in ALS. There is no treatment. Sad thing is, once you're diagnosed, you know your fate because there's nothing you can do about it. So here's cross-section of the cervical spinal cord. And this is the lateral. So basically here we're looking at the it's called the lateral corticospinal tract here. And this is where the alpha motor neurons here in the anterior horn. So basically you get this part and this part affected in ALS. Combined upper, lower motor neuron disease.
Do you know what causes this?
No. We know that there's probably some hereditary. In about 10% of cases, you can find some kind of hereditary link, but the other 90% we haven't had a clue. Okay, if you do the electromyograms, this is an EMG with a patient that has ALS. So ordinarily, when you look at the resting EMG, it should be perfectly silent. When you contract the muscle, you see polyphasic, very high frequency action potentials. And in ALS, see, you're losing motor units. And so what happens is the remaining motor units, the ones that are still alive, will produce these very large amplitude action potentials, and it's hard to, miss that this is not normal. The other thing that's not normal is because you get denervation, you have what's called spontaneous activity. So without any neural control, your muscles kind of begin to twitch. You ever had that, where your muscles just kind of twitch a little bit? You may have early ALS. I just call it fasciculation and fibrillation. Some of that's normal, and y'all are way too young to get ALS. Yeah.
So if people die with ALS after like 4 or 5 years,
Yeah, from the time of diagnosis to the time of death, it's usually about 4 years.
then how is somebody like Stephen Hawking still alive?
Yeah, because he probably doesn't have the more common form of ALS. But if you notice, if you look at what's happening, he's getting much, much worse. You look at the way he was 10 years ago and the way he is today, way different. Okay, but his case is a little different. When you see somebody that actually lives more than 4 years, they probably don't have the prototypical ALS. ALS is probably a wastebasket of a whole bunch of different things.
What is it that actually ends up killing ALS?
They quit breathing. So finally it gets to the point where you no longer have control of your breathing and you die because you no longer can breathe. Last 5 minutes, I want to talk to you a little bit about what biomedical engineers are doing with regards to replacing bad muscles. So we know that we can create these dielectric force components. We can stack them. What we try to do is to get in the kind of the strain pressure diagram. We try to kind of mimic what kind of happens naturally. So we're kind of up in this region here. Piezoelectrics are way too stiff, and magnetic static kind of things are a little bit not stiff enough. So basically we want to kind of get plastic materials that we can activate. And so you can actually stack these dielectrics to where you can create kind of a movement. I might want to read a little bit more about that. The other thing that's really exciting is that we can now create muscles that actually are functional from stem cells. So we can take a pluripotent stem cell, differentiate it into a striated muscle cell or a cardiac muscle cell. And so we can potentially create a natural replacement for bad muscle cells. This holds huge potential for things like Duchenne's dystrophy. And so I personally think kind of this is where the future is. Anyone that gets interested in this, Laura Suggs is actively working on this problem. And so one of the things that we do in our group is we do prosthetics, or we do artificial neuromuscular stimulation. So understand that my brain has control of my muscles, but if I cut the spinal cord, I lose that control. And so I no longer have voluntary control. And so I can replace that with an artificial stimulator. And so one of the cool things here, this is an 8-channel externally powered neuromuscular stimulator. So basically you have kind of the brains of the thing here. They go down to some electrodes, maybe they go to some muscles that are innervated in the arm. You have a little computer, and then you have some way to get a control signal. It turns out that we can do this part really, really well, but we have a little more trouble kind of figuring out what the brain really wants it to do. And so getting a good control signal is actually the weak link in the process here. But if you look at this little device that I have here, which is implanted in the upper chest, if you actually look at the way it works, here's actually the stimulator, and these are little electrodes which we implant in the muscle, and so we can create an artificial action potential, which the muscle cell can't tell. It'll contract when we stimulate it. And so we basically have an RF, a radio frequency control circuit here that we send information in. We send in a pulsing sequence, so we can select which electrode we want to stimulate, and we can also select the amount of current we inject and the pulse frequency. Okay, so we basically have control now over that muscle with these implanted electrodes. So given a pulse sequence that I put into this RF coil here, I will enable my stimulator to send out pulses to these different electrodes, which will cause that muscle to contract in a really a pretty natural way. And so using this, I can get pretty good movement. It turns out that most of your hand function here, your hand is not really controlled by the intrinsic muscles, it's controlled by the muscles actually in the arm. And so with some kind of device like this, I can create a prosthetic device that will allow you to pick something up, voluntary and so on. So this is actually a really cool area to get into as a biomedical engineer. So this is kind of what the electrode looks like. It's a platinum electrode which just slides into and innervates the muscle. Actually, there's even a better way to do it. If you have a spinal cord injury, it turns out that the lower motor neurons are still very much intact. And so what I can do that's even better is I can get some of my graduate students to make these neural cuffs. And what we do is we just wrap that neural cuff around one of the peripheral nerves. So we don't have to stick anything in the muscle. We just wrap this, for example, around the median nerve. And then we have electrodes that come out. We go to the control circuit. What we can do now is we can put the pulses into it's now a denervated nerve. The nerve is not communicating to the brain. But I can now artificially put signals into that nerve and then cause the muscle to contract. The real advantage of this is it requires about 1,000th of the power because we're stimulating the nerve, which then stimulates the muscle. So we get that kind of natural amplification at that myoneural junction. Any questions? All right, see you next time.